Dr. Richard Harris here, clinical advisor to CBD Health Collection, and welcome to our CBD Journal Club, where we review a recent CBD article. And this article was pretty interesting. The title of the article is Attenuation of Oxidative Stress and Inflammatory Response by Chronic CBD Administration is Associated with Improved omega-6 to omega-3 ratio in white and red skeletal muscle in a rat model of high-fat diet-induced obesity. Okay, so high level, 30,000 foot view. What does this mean? Basically, this is a rat study where they made the rats obese and then they looked at giving CBD and what did it do to the ratio of pro-inflammatory omega-6 fats versus anti-inflammatory omega-3s. And we've talked about this on the podcast and on this YouTube channel before, that an improper omega-6 to omega-3 ratio is a big driver of disease. It's associated with heart disease and cancer, diabetes, obesity, insulin resistance, all kinds of neurodegenerative diseases, all the bad stuff. Okay, let's move into the study and the introduction. During the progression of obesity, we know that lipids, extra fat, is deposited in the liver, in our skeletal muscle, and in heart muscle. And this leads to numerous abnormalities. You can detect this in blood cholesterol. You can get elevated blood sugar. You can get insulin resistance, which we've talked about on the podcast before. And this can also lead to chronic inflammation. And this plays a key role in the development of chronic diseases like diabetes, like cardiovascular disease. And what happens with chronic overnutrition, meaning excess calories, this leads to release of inflammatory mediators. We call these cytokines, including a couple we've talked about before on the podcast, TNF alpha, IL-6, and an increased production of ROS, or reactive oxygen species. We've talked about that in the metabolism podcast. What this means is reactive oxygen species, it's an imbalance between reactive species that can damage our cells and the effectiveness of the body's own natural defense systems like glutathione, catalase, and superoxide dismutase, all things we've talked about before, to remove these harmful products. And a high-fat diet provides polyunsaturated fatty acids, which are converted to things like prostaglandins, prostacyclines, leukotrienes, and thromboxanes. These are other messengers that have hormone-like action. And what happens is the omega-6 go towards inflammatory mediators and the omega-3 towards anti-inflammatory. Recent studies have shown that the endocannabinoid system regulates energy metabolism, heat generation, nerve transmission, and inflammation. And the aim of this study was to investigate the impact of CBD on the ratio of omega-6 to omega-3 in different lipids. And what I mean by lipids, fats. Basically, the fats in our body exist in a couple different forms. There's free fatty acids, and then there's a diacylglycerol version, there's a triglyceride version, there's a phospholipid version. All this means is that the actual fat molecules have different proteins or different molecules attached to them, and the body uses these in different ways, as storage, as transport. It's basically just looking at the reservoir of fats in the body, and you can have omega-6s or omega-3s be part of these different reservoirs depending on which type they're in. And so they looked at oxidative stress parameters. We've talked about oxidative stress on the podcast before, and inflammation in white and red skeletal muscle of rats with obesity, and they were given a high-fat diet. Rats really do not like a high-fat diet. They cannot metabolize it well, and this is how you can generate a rat model of obesity. The methods, they used male rats. They had 4 groups. They had an acclimation period of 1 week. And then the 4 groups, they had a standard group, and that's a standard rat chow, which is 12% fat, 57% carbohydrates. Then they had a standard plus CBD. And then they had a high-fat group, which was 60% fat, 20% carb. And then a high-fat group plus CBD. The CBD, they injected the rats once a week, which is not similar to how we ingest it. And the CBD was 99% pure. They collected blood samples for the rats. They looked at muscle biopsies. So red muscle is oxidative metabolism. This uses fatty acids and oxygen. It's also slow twitch muscle. It's called red because it has a lot of blood flow, because it needs blood to deliver oxygen in order to generate energy. And then there's white muscle, and white muscle is anaerobic. It's fast twitch. It uses glucose primarily. So the high fat rats gained weight steadily, and CBD did not affect their weight gain. They wanted to measure intramuscular and plasma lipids, so blood and muscle lipids. And they also looked at several components of oxidative stress, like AGE, which is advanced glycation end products. These are basically what happens when you get too much sugar attached to certain proteins. They also looked at 4-HNE, which is a protein that when we see this, we know that there's a lot of oxidative stress going on. It is a product of when you have oxidized fats in the blood, it can create and oxidize other products, and 4-HNE is a way you can measure that. They also looked at the anti-inflammatory enzymes like catalase and superoxide dismutase. They also measured several enzymes in oxidative stress pathways and genes related to those enzymes. Things we've talked about before: NRF2, the master antioxidant switch. NRF2 also is the gene that codes for these enzymes like catalase and superoxide dismutase and glutathione. And then NFKB, which is the master switch for inflammation. Now the results: obesity caused an imbalance in the omega-6 to omega-3 ratio, so it increased it, and this was improved by CBD administration. Obesity increased the 6-to-3 ratio in red muscle and the various different lipid types that we talked about earlier towards the omega-6. In the white muscle, the glucose, the DAG, and FFA, they actually increased the ratio. Others, the fractions decreased, the phospholipid and TAG. And the authors thought this was meaning that these signals increase lipid storage in this muscle type. DHA, which is a big beneficial omega-3, actually increased. And then also they thought that this might be possibly shifting the profile towards more membrane molecules. So a lot of our fatty acids are used on the outside of our cells and this type of muscle. In plasma, obesity increased the omega-6 to 3 ratio. It increased arachidonic acid, which is the major precursor to the inflammatory cytokines, a lot of inflammatory molecules. Linoleic acid, which is converted to arachidonic acid, and it decreased our omega-3s, ALA, EPA, and DHA. The study found that CBD could help restore this by lowering the ratio and by increasing incorporation of omega-3s into the different lipid fractions. The lipid fractions are the triglycerides, the free fatty acids, the diacylglycerols, and then the phospholipid, those 4 fractions. In red muscle, the omega-6 to omega-3 increase under a high-fat diet, and this reversed when CBD was given, and you saw a big increase in the omega-3s, EPA and DHA. If EPA and DHA sound familiar, that's fish oil. That's what we typically think of. Those are the omega-3s that are in fish oil. There was a large reduction in linoleic acid, which has been shown in other studies to be an obesity promoting factor by increasing inflammation. We just mentioned linoleic acid is a precursor to arachidonic acid, which is a precursor to a bunch of very inflammatory things. CBD shifted the ratio in favor of omega-3s in red muscle tissue, and this tissue are heavily using fatty acids as energy. They also found that the high-fat diet group had higher AGEs, those advanced glycation end products, in white and red muscle, and this was decreased by CBD. And this is a good thing because AGEs are bad. AGEs by themselves indicate insulin resistance. They also deplete antioxidants because we have to use our own antioxidants to get rid of these things. They looked at 4-HNE and MDA. These are 2 products that are caused, again, by oxidized lipids, and these things can damage tissue. This was increased in the high-fat diet, was decreased by CBD. High-fat diet also had an increase in inflammatory mediators like cyclooxygenase COX and lipooxygenase. This was in both the red and white muscle, and CBD actually lowered the inflammatory enzymes here. PPAR gamma, which is a gene involved with energy balance. It's involved with lipid metabolism and glucose metabolism. This decreased with the high fat diet. That's a bad thing. CBD restored normal PPAR gamma in both muscle types. Another thing that PPAR gamma does is it helps with NRF2 and NFKB. So activating PPAR gamma will cause NFKB, the master inflammatory switch, to be removed, and it'll increase NRF2. So what PPAR gamma activation does is it switches us from a pro-inflammatory signal to an anti-inflammatory signal. The high-fat diet increased NFKB expression. That's bad. That's inflammation. CBD lowered that. TNF alpha increased in the white muscle. It was lowered by CBD. The high-fat diet also decreased NRF2 and a gene called BCL2. BCL2 is a gene involved with making sure that cells don't commit suicide. We call that apoptosis, and it's associated with cancer. And CBD actually increased this. So it was making so the cells didn't turn over as quickly. You don't want cells to turn over quickly all the time. That's a bad signal. The high-fat diet also increased two things called MMP2 and MMP9. These are metalloproteinases. What these things do is they break down tissue, and when they break down tissue, they cause inflammation. So this is another inflammatory signal that the CBD decreased. So overall, this study showed that it lowered inflammation in the muscle. It helped restore omega-3 signaling in the muscles. It also increased total antioxidant capacity in the muscles. It didn't change the ratio of catalase or superoxide dismutase or glutathione. The high-fat diet did reduce catalase in the red. CBD reversed that. It enhanced catalase, and it enhanced total antioxidant capacity, and it increased superoxide dismutase in both. So overall, what this study is showing is that the high-fat diet caused significant metabolic abnormalities in these mice. It caused increases in several inflammatory signals, and that CBD was able to help reverse the inflammatory signals and the dysregulation in the omega-6 to omega-3 ratio that was seen with this mouse model of obesity. That's the takeaway. And you can't just generalize this to humans because our physiology is different, but this gives you a insight into how CBD does what it does and why it's one of our favorite holistic products. We actually use the CBD Health Collection products in the house. It is what I use. Otherwise, I wouldn't be out here talking about them, and they wouldn't have sponsored this video. So thank you again for listening to our CBD Journal Club. These videos will come out once a month as I review new CBD literature. Thank you and have a blessed day.